Okay, all right. Welcome everyone. Bienvenue tout le monde. Welcome, where I am just so excited to be here with you all. I'm coming from Waiwaetanong, which is where the river bends, also known as Windsor, and the University of Windsor. My name is Tina Semeniuk. I think you all know me already by now, but I wanted to welcome you to our FishCAST alumni series. And this is going to be our last one of FishCAST. We've been going strong for 6 years. This was always meant to be a 6-year program, so don't worry. It's not like they're shutting us down. We're not canceled. Just like it was only ever supposed to be six years, and so we're winding up. And like before, our whole mission was to try to attempt to train the next generation of scientists in aquatic management and conservation. And as we've known, we don't always end up in terms of where our path initiated us on. So some of us, we were just talking before, are studying mammals now, even though they studied fish before. Some are studying butterflies in national parks. Some of us are studying ungulates. Some of us are still studying fish. And if we're not studying fish, we're fishing. So I think it's just really, really cool to see how everyone has been on their journey through FishCAST and beyond. And today I'm no more pleased to introduce you to Peter Baker, and I'm just going to allow Christian to actually introduce. Christian's also a FishCAST trainee who's been with us almost from the beginning and has had an amazing career and has always participated as well. It's not about you, Christian. I don't mean to be talking just about you. I'm leaving you to talk about Peter. But again, just to showcase just what an amazing troupe of trainees we have, and in alliance and in reciprocity with all the partners that we've worked with, I think it's been amazing. And just setting up your future and just to hear these stories have been really, really fulfilling. And as people would say, my cup filleth over. So with that, just so that we know, Peter's just going to give us a bit of a talk, a presentation, a chat about what he's been up to, what he's been up to, where he's going next. And then afterwards, we're going to turn it over to our trainees to ask some questions. How was it? What were your challenges? Any kind of questions. That's where I'll step back with Kendra and I'll allow all of you to kind of talk about anything you want with Peter. So without further ado, I'd like to introduce you to Christian Tellier. Tellier, please introduce yourself too, to let folks know who you are, and welcome both everybody to our FishCAST seminar series. Thanks so much. Christian, you take it.
Yeah, hi everyone. For those of you who don't know me, former FishCAST cohort, I think 2 trainee. I'm Dr. Chris Terrian now. Happened 2 weeks ago. I'm currently the protected areas biologist with the Ontario Ministry of Environment, Conservation and Parks with Ontario Parks. And I'm here to talk to introduce my good friend and former lab mate, Peter Baker. Peter currently is a fish and wildlife biologist with biodiversity pathways at the University of British Columbia. He received his master's in science from the University of Western Ontario in 2023 under the supervision of Dr. Brian Neff. And there he was a trainee in the second cohort of FishCAST. His research focused on the cumulative effects of invasive species and climate change on the cardiac physiology of lake trout in the Great Lakes and in inland lakes. Peter's research interests lie at the interface of anthropogenic ecosystem change and animal physiology across both aquatic and terrestrial systems, with an emphasis on collaborative research with Indigenous communities and the co-development of knowledge. As I mentioned before, he's a good angler, he's a good bushman, he's got a lot of great stories, had a really interesting career path, and I think you all will learn a lot about him and look forward to the question period. And without further ado, I'd like to present to you Peter Baker.
Well, I love it. Thank you so much, Chris, and congratulations on the new doctor title. That's awesome and very well deserved. I will say you were a fantastic mentor, and I wouldn't be the bushman and fisherman without the knowledge that you shared, so appreciate it. It's fantastic to be here. Thank you everyone for coming. It's awesome to be here as a FishCAST alumni. It's really great to see some familiar faces. It's great to see some unfamiliar names as well with people I haven't met and worked with yet. This feels like very much a full circle moment for me, coming back after years of being out of, well, mainly the fisheries scene also, but just out of FishCAST, so it's great to be back. I'm excited to talk to everyone today and present on the journey that I've been on through my master's degree, through FishCAST, through my career as a biologist so far in the years after school. And I titled my talk today, You're Not Stuck, A Biologist's Winding Path. And a primary theme of my talk today is about how career path in biology is rarely linear, as I'm sure you've heard from other FishCAST alumni seminar series. And mine, as you'll see, is, like others, very far from linear. And I thought my career path was going to be very linear in grad school. So this came as a big surprise to me, but I'm rolling with where the career path goes. And a big part of the message today is, if you're feeling stuck, to really just embrace the nonlinearity. You don't need to stay where you are. You don't need to study the same thing that you studied in grad school, even though you spent years becoming an expert on that specific topic. And really following the evolution of your curiosity can be extremely rewarding, which I hope can become clear throughout this presentation. And as some of you know, we were just discussing it, but I moved to BC after grad school, and it is just absolutely nuts out here. I'm blown away every day that I get to spend outdoors here. So I tried to incorporate as many relevant photos and sometimes non-relevant photos that I could throughout this talk. And many of them I actually took while I was working out in the field, like this one on the title slide here. This was at one of my field sites for a camera trap program that I was running or helping to run. And that's just one of the rewards for following your curiosity, which I hope you'll also find more rewards of that throughout this talk. So hopefully slides are progressing here. Let me know if I'm talking in slides or not. So as I mentioned, today I wanted to touch on a number of things. I wanted to briefly touch on my journey through grad school, my master's research, my journey after, or highlights with my time with FishCAST and journey after academia as well. Some of the projects that I'm working on now, which are not fish related, and then also some chapters that are coming up next. But before I get to where I'm at now, for context, I want to take us all the way back to the very beginning for me, just really quickly. I grew up in a small town about 3 hours north of Toronto, and right from the get-go was really fascinated with everything about nature. I'm sure like many of you, I've got memories from when I was a kid, picking up every creepy crawly, every salamander, every beetle, really touching the most gross things that I could possibly touch. And as I got older, that never really stopped. In fact, the diversity of my interests kind of grew even bigger. And I can truly say that I'm a bit of a pure generalist when it comes to my fascination with nature, and with no clear path on a particular like animal or group of animals that I'm mainly interested in. And during my early career, this became evident with the variety of jobs that I ended up taking on through high school and through my undergrad. I worked with everything from birds, bees, freshwater mussels, mosquitoes even. And I really just wanted to experience everything and do it all. But if I had to give a slight edge to a particular group, it would be fish. And fishing was really the one thing that I couldn't do without growing up. Like I could go a whole year without collecting insects in a field or looking at moths at night. But I would never in a million years miss a spring pike fishing trip in Georgian Bay. And this is what led me to pursue a Master's of Science in Fisheries, which started back in 2021 at Western University with Dr. Brian Neff. And my research looked at the impacts of dietary thymidines on the cardiac physiology of lake trout. And I'm sure many of you are probably already familiar with, at least to some degree, the issue of thymine deficiency, either from your own research or hearing myself or Chris or Kevin Adeli, who's also FishCAST alumni, who we all worked on similar things. You've probably heard us talk about it at some point. But for those who don't, or who don't know, who are not familiar with thymine deficiency, this whole issue stems from two particular prey species, alewife and rainbow smelt, which are pictured on the slide on the bottom left. And they're not native to the Great Lakes, but they are now particularly abundant. And these species, when they're consumed by predators in large enough quantities, they cause a deficiency in vitamin B1, which is also known as thymine. And this is thought to arise because these prey species have high concentrations of an enzyme called thyminase, which breaks down thymine when predators eat a ton of them. So essentially what we did, or what I did in my master's and what Chris did also, is we raised a bunch of lake trout in our on-campus hatchery, which you can see a photo of in the top right corner of the slide. And we fed half the fish a control diet and half the fish a diet that was loaded with that enzyme that causes thiamine deficiency. And what's really interesting to me is that thiamine deficiency is not just a fish problem. It's also a problem in people, and it has been a problem in people for thousands of years. And when people get thiamine deficient, one of the main symptoms is cardiac related issues. And when I was starting to think about what I wanted to do for a master's degree, I recognized that nobody had looked at this in fish yet, and in my head, it was, if people can get it, why can't fish? And so that's what I looked at. And so essentially, we raised these fish in our on-campus hatchery for 9 months, and then I essentially performed fish echocardiographs on their heart and looked at a number of things. And here's some snippets of what the echocardiogram outputs are. The photo in the bottom right is me actually doing the echocardiograph. And essentially what we do is we anesthetize these fish. We put them in like a half cut open PVC pipe just to hold them in place. And it makes it easy to get some measurements on their heart. And whenever I'm presenting on this work, I really like to show what these heart rates sound like and look like, because I think they're really cool and it's really interesting to do. So essentially, I looked at heart rate across a range of temperatures. And since heart rate is a key determinant of thermal tolerance, I thought that if thiamine deficiency was indeed messing with their hearts, it might be impacting their ability to tolerate warm water temperatures. And so this is what a baseline heart rate would look like, and that would typically be around 9 degrees. And hopefully everyone can hear this sound associated with it. So pretty nice, relaxed, steady heart rate. And if we start to increase the temperature, heart rate increases exponentially until it peaks. And that peak would typically occur around 17 degrees Celsius, 70 to 20. And this is what a peak heart rate would sound like. So it's really pumping. And then if we keep increasing this temperature up, eventually we reach what's called an arrhythmia temperature, where the heart rate or heart function essentially stops. And this would typically occur around 20 to 23 degrees Celsius. And this is what an arrhythmia temperature or a heart rate at the arrhythmia temperature would sound like. So we get some heart function, some dysfunction in there. And then after doing this with dozens of fish, I found that when we raised these fish on the diets with thiaminase, that they had noticeably larger hearts was a big thing that I found. And what's really interesting is Kevin looked at this further for his thesis, Kevin Adeli, and found that these hearts were actually getting damaged and quite severely damaged, which was causing them to be really large. And because their hearts were getting damaged, I found that they weren't functioning as properly. So essentially, they couldn't reach the same performance level that the control fish could. So lower heart rates at their peak temperatures. And because their hearts weren't functioning well, they had a lower thermal tolerance. So they couldn't tolerate those warm water temperatures. And the arrhythmia temperature was occurring far earlier on the scale than the control fish had it. So I thought that was a really interesting connection between invasive species and climate change, like maybe a bit of an obscure connection, showing how those two stressors can interact to negatively impact lake trout. And so that was my research in a nutshell. But while I was hunkered down in a lab with no windows doing those trials, I was also having an absolute blast during my FishCAST internship, which I would say is probably the highlight of my graduate degree. And that was all made possible because of the FishCAST program. And so I did my internship with the Ontario Ministry of Natural Resources based out of Owen Sound. And I was able to take part in a really wide variety of projects. I was really thrown into a little bit of everything. I got to do work tagging rainbow trout as they migrated upriver at Denny's Dam in Southampton. I got to go out on Georgian Bay and use trap nets to catch absolute monster muskie and tag them and just be in their presence, which was phenomenal. And I also got to do a project with channel catfish, which are the photos on the bottom right and top in the middle. And it was just amazing to see some of the Goliath sizes of these channel catfish. And you can see by the expression on this biologist's face just how cool these animals were. And so this internship really gave me the opportunity to get my hands dirty and learn many of the different field techniques that were actually used by fisheries managers. And this was critically important to me as I didn't get to have those skills or experiences during my actual master's work. And on top of all the things that I learned out in the field, I made some just amazing connections with the biologists and the technicians that I worked with. Many of them I still stay in touch with today, and just really, really great people all around. So a big shout out to the Owen Sound field office. You guys are awesome. And on top of that, like, you can ask anybody in biology or any field for that matter. It's all about making and maintaining connections. So that's what's really going to drive you through a career. That's what's going to help determine a career path. And that's what this internship really allowed me to do. And on top of that, another FishCAST highlight for me was the micro credentials. And I completed, I think, around 10 of them, and they were all phenomenal. But the one that stood out to me was the outreach micro credential. This was something I had very little experience with as a scientist, but I was really interested in. So I decided to try writing an article for Ontario Out of Doors, which is a hunting and fishing outdoors magazine in Ontario. And it's a magazine that my dad would always have around the house growing up, and I would always just, pick up and read. And so I figured I would try writing an article to get it published. And in July of 2023, it happened. It got published in their July issue. So that was an awesome experience, and I learned a lot about what it means to communicate complicated scientific topics to a general audience. And this is something that I wouldn't have done on my own if it wasn't for FishCAST giving me the platform to do so. So that was great. And that was 4 years in a nutshell, very quickly. And that brings me to my first major curve of my very nonlinear path as a biologist so far. And so after grad school, I defended in June, and then in August, I moved out to BC with my now wife and as I got a job as a fish and wildlife manager for the Adams Lake Indian Band. And the Adams Lake Indian Band is one of 17 First Nations communities within what's called the Secwepemc territory. That's pictured on the left-hand side, which takes up a really big portion of kind of central, eastern, southern area of BC, encompassing like Kamloops and Revelstoke and going over to Banff and Lake Louise and that area. And this was a huge leap for both of us. We had no friends, no family, no connections out here whatsoever, had never really even been to this area, but we did it anyway. And so we moved to Kamloops in the fall of 2023 and really spent the first couple months just trying to soak in this territory and really just get a lay of the land. And we were blown away with what this territory looked like. Within a 3-hour drive, we could see the tallest mountain in the Rockies. This is Mount Robson in the bottom left-hand photo corner here. We could see grasslands, deserts, rainforests, and it's just next level. It's amazing. But when we moved out to BC, it was literally on fire. This was one of the worst wildfire years on record, I believe. And there was a particularly big wildfire right where I was working. And so this picture on the bottom left is Adams Lake, a 5-minute drive from my office. And I didn't take this photo, but I did take the 2 photos on the top that are of the aftermath. And so you can actually see the lake at the bottom of where this fire is, and the whole hillside is just completely in flames. And this is a tiny, tiny fraction of the land area that was burning. And so this fire in particular planted a bit of a curiosity seed that I'm going to come back to near the end of the talk. But it was just a huge eye-opening experience. Growing up in Southern Ontario, never really experienced what wildfire was or what it did to a landscape. So this was amazing to see. So most of my job, right from the get-go, was trying to learn what impact that fire had on all the culturally important species for the Adams Lake Indian Band. And a big part of this work was working with fish, which was awesome. That was my wheelhouse. And this included looking at how the wildfire impacted kokanee spawning habitat. So the picture on the left is us catching kokanee in the river and doing some measurements on them and seeing how their spawning success is. Some of our projects involved looking at sockeye, tracking smolt out migration, and so looking at their condition on their way out to the ocean. And another big project that I was leading was actually taking the skills that I used during my FishCAST internship, which is where I learned how to gill net for the first time, and leading a communal fishery for the community. So we'd essentially put out gill nets and catch salmon that we'd take back to shore, and we'd get the youth out, the elders out, everyone would fillet fish together. There'd be knowledge sharing, smoking of the fish, and it was just a really cool experience to learn how these salmon are being used culturally in the community. But what I loved most about this role was that it wasn't just about fish. This is really where I got my feet wet with the world of wildlife management. And I did a little bit of everything in this job. I got to do work doing breeding bird surveys, so looking at how the impacts of that wildfire had on bird diversity and bird abundance, doing species at risk survey with bighorn sheep. But a really big focus of this was with caribou. And that brings me to my second big curve in the nonlinear path. And so it was this work specifically with caribou that led me to pursue a job with Biodiversity Pathways, which is who I'm with now. And Biodiversity Pathways is a not-for-profit based, we have people kind of all throughout BC, all throughout Alberta, a couple folks in Ontario as well. And we're affiliated with UBC, University of Alberta. And a big part of what Biodiversity Pathways does, or especially our little subunit called the Wildlife Science Center, focuses on caribou. And apologies to all the fisheries and aquatics fanatics listening, but I'm going to pivot away from fish for the rest of this talk and discuss what I've been working on for the past couple of years, which has been primarily focused on caribou. And for those who don't know caribou well, like I did not know caribou at all, living in Southern Ontario. You might be surprised to know that they are found literally all across Canada, everywhere from the Yukon all the way out to Newfoundland. And historically, they would have been found in a lot more places. They would have been found as far south as like Idaho and Montana, all the way down into the maritime provinces and central Ontario. And there's a number of different kinds of caribou. And they differ, across Canada. But the primarily group of caribou that we work on is southern mountain caribou. So this little group in southern BC that are the pink herds, central BC, kind of down to the American-Canada border. And southern mountain caribou are not doing very well. There are 32 different herds or subpopulations of southern mountain caribou, but in the last couple decades, 15 of them have become extirpated or functionally extirpated. And there are roughly half as many caribou on the landscape as there was in 1990. And it was this really sharp decline that piqued my interest as a biologist to try and figure out the intricacies of why they're declining. And I quickly realized that the answer to the question, why aren't southern mountain caribou doing well, wasn't really that hard to answer at a surface level. One look from satellite imagery of an area where caribou used to be will give you a pretty solid idea of what's going on. So this is a zoomed in satellite photo of my neck of the woods. So down the bottom left-hand corner is Kamloops. Kind of in the central area is Salmon Arm, and then I don't have them labeled, but like Revelstoke is up and over here, and then Banff and Lake Louise would be over on the right-hand side. If you can see my cursor, hopefully you can. And I live in a small town called Blind Bay, which is kind of on Shuswap Lake, a little bay over here. But essentially where I worked for many years was up and around Adams Lake, which is this one here, and up and around this area. And this is where caribou used to be. And a lot of caribou used to be in this area. But now caribou are mostly confined to the lesser disturbed areas of the mountains. And you can see, just without even zooming in on this slide any further, you can see around Adams Lake, it is just a checkerboard of logging in the area, from cut blocks and other disturbances like mines. And it gets even crazier when you consider all the roads in the area. Now, I moved out to BC thinking it was going to be one of the most remote landscapes that I have ever experienced. And, as far as Southern Ontario standards go, it is very remote, but there are very few areas that are untouched by humans. And so this is a picture of the roads in the area, and it's just unbelievable. And I've heard it said that there are enough roads in BC alone to wrap around the Earth 40 times. So there are very few places that you can't access. But it's interesting, why does this disturbance actually result in decreasing populations of caribou beyond the obvious habitat loss from the cut blocks and the roads and those sorts of things? So this is something that my team has been working on for many years, and it's quite in-depth and quite interesting. So what's happening is that from these disturbances, such as logging, but also like natural disturbances like wildfire and pest infestations, we get a lot more young forest on the landscapes. So you can imagine we have a cut block go in or a big area that's clear cut. We've got a lot more new growth coming up or early cereal vegetation. And that early cereal vegetation or like young leafy plants are really good news for moose and the deer. So the moose and deer populations start to do really well in these areas that are heavily disturbed. And when you get moose and deer populations doing really well, so do predators like wolves, like cougars, and a number of other predators that we have on the landscape out here. And when the wolves do well, that's bad news for caribou as they get preyed upon at a higher rate. And this sort of indirect impact that moose and deer have on caribou is called apparent competition. And on top of that, just when you thought caribou weren't getting enough from the wolves, some of the work that my colleagues have done over the past couple of years discovered that the predators are actually using the linear features that we're putting on the landscape as highways. So they're using them to hunt, to travel faster, to travel further. So they're using the roads, they're using the pipelines and the seismic lines to get at the caribou. And when they do this, they cover more ground and actually increase their hunting efficiency on caribou. So landscape disturbance is really doing a number on caribou from multiple different pathways. So what are the different things that we can do about it? And there's lots being done about it right now for southern mountain caribou. One thing that we can do is protect the caribou themselves. And this is being done using what's called maternal penning. So that's bringing in pregnant females into a protected enclosure to increase the survival of the offspring. And that works in some cases. Another thing that we can do, and that is being done, is tackling this whole apparent competition issue. So managing the wolves, managing the moose, and managing the deer, that's being done in a number of herds. But both of these things are really just short-term solutions. If there's still disturbance on the landscape, we're going to have to keep managing the wolves and everything else. And if we really want to fix the root of the problem, we got to get at habitat. And so this has been a focus of my work over the last couple of years, is fixing the habitat issue. So this is an example of some road restoration work that I led back in 2024, where we basically were trying to make the roads in caribou habitat as hard to walk on as possible for the predators. In fact, we would make it so hard to walk on that it was easier to walk in the bush beside the road than it was to walk on the road. So this project basically involved getting a backhoe out into caribou habitat and just shredding the road up as much as possible, making it absolutely impossible for any predators to walk along it, or anything for that matter. And you can really see the difference, especially in the photo in the top right-hand corner. I like to use this example. Nice, easy to drive on road, and then you can see where our treatment begins there. And we really just shred it up. We throw all the debris from the side that we can on it. And I can speak from experience that it's really hard to walk on. And we test it. That's how we test in the field, whether it's a good treatment or not, as we try to walk across it. And this is a video of that same treatment. I basically just put a drone, hopefully it's not like laggy or anything for anyone. But yeah, put a drone up in the air to get a better aerial look at this treatment. And you can really see the difference between where the road is and where the road no longer is. And so how do we know that all of the things that are going on for caribou are actually working? You know, we do all these things. We protect the caribou, we manage the wolves, we manage the deer, the moose, we fix the habitat. How do we know what those are actually doing? And so another thing that my team has been working on is trying to figure out ways to monitor the impact that all these treatments have on the ecosystem. Um, and like beyond just wolves and moose and deer, how are other animals responding to these treatments? So to try and answer this question, over the last couple of years, my team has deployed nearly 300 remote trail cameras in a really big remote camera grid that spans 16,000 square kilometers, with including national parks in other areas. And we're in collaboration with many different First Nations communities in Parks Canada as well. The photos on the right are some photos from a couple field days back in September. A couple of them required us to hike in off the road, and you can see I'm very happy to have our camera deployed in this photo on the right. It was a long, long hike in, so I was happy to have that one done. And a lot of the sites we could get to by road or by hiking, as you can imagine, after I showed all of those roads that we have in the area. But some of them we could only get to by helicopter, and that made for some really awesome days in the field, especially out in the mountains. So the photo on the left is I was in the back seat of the heli on our way to our field site. Beautiful bluebird day, just some awesome views of the mountains. The photo in the middle is us training some Adams Lake Indian Band Guardians on how to deploy the camera, and how to do it to standardized practices. And while we were out one day in the field in September, we actually came across, a decent sized group of the endangered southern mountain caribou. And it wasn't a huge group, but it wasn't that small of a group either. And afterwards, I was looking at the photos I took, and I realized that this group we saw represented 10% of the entire herd in the area, which was just, goosebumps being able to just experience that and see them. And we also get some just really great photos from these cameras, and I wanted to showcase a few of them. And this was one camera that we left out for 2 months. We had to go back early and service it, but I was, just itching to see what it got. And this particular site just had incredible diversity on it. Like, you name an animal, it was on this camera. Everything from like moose, wolves, bears, skunk, raccoons, pine marten, everything. And so really just, animals that you wouldn't know were there at such high diversity if you didn't have a camera looking. And over time, what this project hopes to do is we collect enough data from these photos to actually be able to estimate and monitor animal density over time, with a particular focus for caribou, which would be looking at the wolves and the moose and the deer to make sure that their populations are responding in the ways that we would expect them to with the different treatments that were going on in the landscape. And this is really important to be able to monitor the effectiveness of those different recovery techniques. So that, in a nutshell, is the story of southern mountain caribou. And you might be wondering, how are the other caribou doing in Canada? Well, my focus actually at Biodiversity Pathways in the last couple of months has shifted to, instead of focusing on southern mountain caribou, focusing on northern mountain caribou. So northern mountain caribou are these red herds. I can't see my cursor, but hopefully I'm in the right area. The red herds spanning from central BC all the way up to Yukon and the Northwest Territories. And in general, they're doing much better than southern mountain caribou, probably because there's far less disturbance in those areas in general, but they're actually really understudied. And in a lot of cases, we don't actually know how they're doing. So I just got back from a trip up to the Northwest Territories. I just got back like a week ago, studying the Redstone caribou herd, which is right on the border of the Yukon and Northwest Territories. And this was the first time that this herd had a composition survey, which is looking at the ratio of cows and calves and bulls. This was the first time it was surveyed in over 20 years. And here are some photos and some footage from my time up in the Northwest Territories. This was an incredible experience. It was one of the most remote places in North America by far. Very little human influence in this landscape. And I was very surprised. What might seem like a barren winter wasteland was actually just teeming with wildlife. Caribou all over the place, wolves, moose, wolverine, ptarmigan. And it was just a really awesome experience. And we have a pack of wolves in the photo on the top there. They just look like ants, but I promise that they're wolves. And so that's been my role most recently at work. And it seems like a very far reach from my fisheries days only a couple years ago. And to top it off on my very nonlinear path, I'm starting my PhD this fall at UBC, studying mule deer. And remember when I said that wildfire that was burning in BC when I first got here planted a curiosity seed? Well, this is it blooming. So my PhD will be looking at the impacts of wildfire risk reduction. So that would be like commercial thinning, cultural burning, seeing what impact that has on mule deer habitat selection and physiology in the Caribou region of BC, which is around Williams Lake and Quesnel in central BC, for those that know. And this past winter, I actually was able to get out and start trapping and collaring deer for my project. And that's what these photos are from. This was my first ever collared deer for the project, which was very, very proud and very important moment. And this is kind of a photo of the traps that we used to get the deer and one of my lab mates setting the trap up. And so that brings me to where I am right now on my ever so winding path. And there's a few things that I hope are clear take-home messages from this experience. First off, I hope what's most clear is just follow your curiosity. Don't ignore it. Don't be afraid to go outside your comfort zone. I guarantee it's going to be the most rewarding thing that you can do. And second, what was really important for me to learn was that a door open does not mean a door closed. Just because you decide to pursue a different career path, it doesn't mean the other path is lost. You still have all the skills and the experiences you gained, and you can always go back to that path. And lastly, embrace the non-linearity. This was something that was really hard for me to embrace, as I envisioned my path to be linear. You don't need to feel stuck. It's what makes a career in biology exciting. And you just have to always remember and tell yourself that you're exactly where you're supposed to be. And that's it. Thank you so much, everyone. This has been a pleasure. Happy to chat and take any questions.
That was awesome, Peter. Thank you so much. I'm just going to turn it right away to our audience. I'm just going to take a moment, absorb it all. I think it's amazing. And I love your biodiversity pathways and wildlife science center. Those are really cool. Oh, I think there's new messages coming in. Yeah, very super cool stuff. Any questions for Peter? Because I don't want to dominate the question period. I'm going to leave it up to you folks, but.
I also can't see the chat, so if anybody has something in the chat, let me know.
Okay, we'll do that.
Tina, do you want me to stop recording?
Oh, yes, please. Thank you.
Okay, before I stop recording, I just want to point out to Peter that like the slide that worked during practice with like the heart sounds didn't work when you were talking, but I didn't want to interrupt you. So I don't know if you want to go back and try to play it again.
Yeah, let's do that.
Yeah. I was going to jump on, but then I'm going to disrupt your flow.
Oh, I might. Oh, shoot. You know what happened is when I reshared my screen, I didn't reshare it with sound. That's what happened.
Okay.
It's okay. We don't have to share it.
Okay.
The visuals were so clear. Like you can definitely see. Oh, yeah, that's a review.
Yeah,